So before I get into this talk, I'd first like to thank the JGI and NSERC for jointly funding this project through the FICUS program. And I'd also like to thank NERSC, Compute Canada, the Tula Foundation, and Ecoscope for their continued support. Additionally, I'd like to thank Connor Morgan Lang for creating the TreeSat pipeline. And I'd also like to thank Monica Torres Beltran and Elise Hawley for their continued work on the distributed nitrogen cycle, as well as my supervisor, Stephen Hallam. Finally, I'd like to thank our colleagues at the University of British Columbia, as well as our distributed network of collaborators. So today, I'll be going over the basics of the distributed metabolism and how that looks across a redox gradient, with particular focus on the nitrogen cycle. Then finally, I'll be talking about how we can use the functional anchor genes found in these redox gradients to perform fast phylogenetic mapping using the TreeSat pipeline. So, as you're all aware, we live in a world that's dominated by microorganisms that occupy virtually every ecosystem on the planet. And these integrated communities facilitate the mass and energy transfers, which both support and shape our microbial communities through distributed networks of metabolite exchanges. Now, what does the distributed metabolism look like within the context of a microbial community? Well, at the individual level, you could have an individual taxa that has all of the genes that encode each step of the metabolic pathway encoded right into its genome. Alternatively, at the population level, you could have one population conducting an entire metabolic process, but as you can see here, the genes start to get distributed within this population. And then at the community level, you could have each gene that facilitates each step in a metabolic process being distributed across different taxa and populations within that microbial community. So given this context of a distributed metabolism, and given the fact that we know that environmental constraints shape microbial community structure, we can start to think about how these environmental conditions can also shape the distribution of genes within these communities. And we can also start looking at that within the context of a redox gradient, which can be found within oceanic oxygen minimum zones. Now, OMZs are fairly important because they comprise of about 7% of the world's oceanic volume, and they reach concentrations dipping below 20 micromolar. On top of that, these OMZs are expanding due to increased surface temperatures of the water. As you can see here, there are many study sites of OMZs all around the world, and our lab focuses particularly on Saanich Inlet, which is a seasonally anoxic fjord found off the coast of British Columbia. So we've been collecting both chemistry and multi-omic data at Saanich Inlet once a month, every month for the past decade or so. And as you can see here, the oxic and anoxic layers shift up and down the water column as the temperatures increase and decrease with the seasons. Also, if you look a little bit closely at points closer to 2007 and compare them to points at 2014, you can see that the overall concentration of oxygen in both the oxic and anoxic layers has been decreasing over time. And also, you can see that these anoxic layers toward the bottom of the inlet have also been increasing. Now, what makes seasonally anoxic fjords different from other open ocean OMZs is the fact that they become so anoxic at the bottom. So as you can see here, you have the consumption of oxygen going all the way down the water column, and as you become anoxic and anaerobic, you start metabolizing nitrate, as you can see right here. And with the consumption of nitrate, you will also start going down the redox gradient of terminal electron acceptors until you start metabolizing sulfur and carbon into methane gas. So given the fact that we have this redox gradient here, we can focus a little bit more specifically on the denitrification pathway here. So the nitrogen cycle is fairly important within this system because it's responsible for approximately 50% of the ocean's nitrogen losses through the production of nitrous oxide. And as you can see here a little bit more closely, we actually know the genes that are responsible for facilitating each step within the denitrification path. So given this information that we have, and we start linking that back to this idea of distributed metabolite exchanges, then we can start thinking about how different members within a microbial community can metabolic couple with each other in order to share and process different metabolites within this context. So fortunately, we happen to have phylogenetic trees that are centered around each of these functional anchor genes. And on top of that, we have a fairly good idea as to who is there and what they're doing. But there may be other members that have these genes that may not necessarily be apparent to us at that time. So that leads me to the TreeSap pipeline, which provides a more phylogenetic context for determining which taxa are involved in this distributed process. And through this taxonomic assignment, we can start to piece together the nitrogen cycle within the context of distributed, excuse me. We can start to piece together the denitrification cycle based on the taxonomic units which are responsible for each step within a given metabolic process. Now, TreeSap is a tree-based, sensitive, and accurate protein profiler, and it works by feeding it any kind of genomic sequence. It'll then convert that genomic sequence into an amino acid sequence, and then align that sequence to your reference tree. And based on that alignment, it will then produce both a classification table as well as a file that is readable by the interactive tree of life. Additionally, you could also tell it to update the tree if there are other taxa that it hasn't placed within this phylogeny. So in terms of the data that you can give it, it can be anything really. It could be an isolate genome, a single-celled amplified genome, a metagenome assembled genome, or a metagenomic context. But for today's talk, I'll be looking specifically at metagenomic contexts that were collected on August 2012, and these samples were collected all the way across the water column, starting at 10 meters, going all the way down to 200 meters, all the way down the redox gradient. So we also ran this pipeline on all of the 7 genes that are involved in the denitrification path. However, I'm a little short on time, so I'll only be focusing on NAP-A because of its prevalence throughout the water column. And there's some fairly interesting results to see there. So, looking a little bit more closely at the taxonomic placements that TreeSap has conducted, you can actually see where the different proteins were mapped onto the tree of life. The size of the dot that's on this tree maps back to the number of proteins that were mapped onto these branches. Now, if you look a little bit more closely toward the root of the tree, you can see a couple more dots. These are dots that didn't necessarily get mapped into the taxonomy that was provided. So what's nice about TreeSap is that you can actually update the tree with these novel sequences and produce information on that as well. You can also feed it metatranscriptome and metagenomic abundance data. And this is fairly interesting because you can start to see the cast of characters that are both present in the microbial community at these different stages, and you can see what their transcription looks like. Now, the outer ring represents 10 meters, and the closer you go down to the center of the tree, you go further and further down the water column. Now, if you look a little bit more closely, you can actually start to see differences in the community structure based on the metagenomic abundances here, as well as differences in the transcription. Now I'm going to focus a little bit more on the 200-meter sample. And in this case, there's a few interesting things that you can see here. So there's some fairly expected cases where you have low abundance taxa with low transcription rates, and you also have some fairly high abundance taxa with very, very high transcription rate. This is a group of gamma proteobacteria, which is fairly well known for its contributions to denitrification. But you also have some instances where you have very low abundance taxa, particularly in this case, it's delta proteobacteria, that also have fairly high rates of transcription, which is a fairly interesting result. So this is actually a summary slide that encapsulates all of the different genes that were run at all of the different depths. This is a figure that is literally hot off the presses. I got this figure this morning, so I'm really excited to share this with you. So what can we see here? Well, if you look closely, you can actually see that this process is very highly distributed because you don't have a single taxa that has all of the genes that perform the denitrification process. Also, again, you can see different taxa that have very, very high rates of transcription, but are not very abundant within these areas. Also, you can, if you look a little bit more closely at NOR-B, you can see taxa that are fairly abundant and also fairly highly transcribed. So based on this information, we can start to think about the different taxonomy that are involved. We can start to think about the different taxonomic units that are involved in each of these different steps within the nitrogen cycle. And we can start to think about reconstructing the nitrogen cycle based on the different taxa that have the genes that are involved in each step within the nitrogen cycle, and potentially look a little bit more closely about at the ones that are transcribing those molecules. That concludes my talk. I encourage you to find Connor Morgan Lang and ask him questions about the TrueSat pipeline, particularly the nuts and bolts of its functioning. And I'd be happy to take questions at this time.